Hi. So let's continue the first notebook. Let's continue where we stopped. So this is the course web page. When you go here to the code notebooks, we are in the first notebook. These are the slides, and then we're gonna go to the part 5.2. That's the visualizing a GWAS. So a GWAS is a genome-wide association study. The most simple definition of a GWAS is that it is a statistical association between a phenotype, right, a trait, and a genotype. This association can also be called a biological association. So the figure that we're going to generate is based on this GWAS 2019 paper on Huntington's disease. I can show you. This paper is in the drive of the course. So this is the bioinformatics bridge course drive. There's this papers folder there, and then I click on the GWAS 2019 paper. The title is Mutant Huntingtin Affects Diabetes and Alzheimer's Markers in Human and Cell Models of Huntington's Disease. So when I scroll down this paper, we are going to see in figure 3 that there are a couple of genes here that we are showing the genetic association or the association of the genotype. That means the SNPs. SNPs stand for single nucleotide polymorphisms. Each one of these dots is a position, right? So in the x-axis, you can see here, for example, in this third plot here, this is also called a Manhattan plot. You can see in the x-axis that this is chromosome 4 position, and then in the y-axis is the minus log 10 of a p-value. I think we discussed what a p-value is. It measures a statistical association, okay? So what we wanted to show in this paper was the association of diabetes marker genes, these sort 1 through 4 genes are, they're called sortilins. These are genes that are that are known to be associated with diabetes, and we showed that it was also the case that these diabetes genes were associated with Huntington's disease. You can see the dots colored in green here. These are the positions, okay? These are the SNPs that have significant association with Huntington's disease. So let's take a look at how we can do that in R, okay? So again, we are doing a visualization of this genome-wide association study. We are going to use this library called QQman. So if you do not have the QQman library, you can install it. I'm gonna come here to the Launchpad, and I'm going to open the R console. I'm gonna put it here. So I'm gonna type here library QQman. I'm gonna see what happens. There's an error. I just downloaded a more updated version of R this morning. That's why it might ask me, I might have to install this package. So it's not there. It means I need to type install.packages, and then I'm going to open parentheses, close parentheses, QQman. I think this symbol stop started here, meaning that it's running something, not something, it's installing the library. I did not know it would take this much long. Let me see if I can do it in RStudio. Let's open RStudio. Was faster in RStudio. I'm gonna stop this. I'm gonna stop the R console. I'm going to say do not save it. Let's see if I can, because I installed it in RStudio, I assume it's installed now in the R console, which means in R. So let's try again library QQman. And if oh, should not have done this. I wanted to just do the library QQman. Okay, so it's installed in the R console now. It's also installed in the RStudio. I'm going to continue in the R console for the sake of simplicity. Let me see, for example, usage run, please run vignette QQman. For example, usage, please run blah, blah, blah. So this message is the same as this message here. Okay, after we install or after we load the QQman library, we are going to load the data frame to be visualized. In the previous session, we discussed the concept of a data frame. Remember, I was showing the iris dataset, for example, and we were discussing the columns and the rows in that one data frame. So now what we're going to do with this command here is we're going to load this GWAS table, and it's going to be this chr4.txt object. It's basically a text file. You should be able to find this file also in the bioinformatics bridge course, and then in the data folder. Okay, there you go. Here is the chromosome 4 data. So let me go back here. So this is what the object is. I need to do one thing. I need to set, this is important for you guys. What I have to do now is this console that I have open right now does not know where in the computer the file is located, okay? And that's going to depend in your guys' computers where you're going to save the data file, okay? It so happens for me that I already have the text file in a folder called data, of course, and it so happens that I know in my mind where in my computer that is located, and so now I'm going to type what is called a path. So R knows where in my computer I want the console to look at. So what I'm gonna do, I'm gonna type this command set working directory, and I'm going to open parentheses and close parentheses. I'm going to do open quotes, close quotes. So my chromosome 4 data should be in my desktop. And then there is a folder there called Gipoliano. I need to go to a folder called Recombio Scientific. And then there, no, actually, That's not where I want to go. I want to go to the bioinformatics bridge course. And then it's a 2024. It so happens that I have two, but it should be this one here. And then where is the data folder? Seems like it's not this one. Actually, let me, it's better if I open it here. So the bioinformatics bridge course, it's this one. It should be this one. It's not going to be here. It's actually in the recombile. It should be this one, recombile, and then 6 neuroblastoma, and then data. So this folder data there has the same name as the one that is in the Google Drive, and that's where we should find the, man, it's, no. Where is it? I have to, chr.txt. chr4. .txt. Okay, it's in the recombile tutorials. Where else? It's in the one introduction. Okay. Recombile scientific, recombile book English, recombile, and then one. Yeah, because it's the introduction. Yeah, we are talking about the introduction. Okay, so that is the folder. So set working directory, it should be that one. Okay. Now we can type this command. I'm going to create this object GWAS underscore table, and I'm going to use the assignment operator. Read, I'm going to type this function, table, open parenthesis, close parenthesis. So we have already set up the working directory. That's why we should be able to get the CHR file there. And I'm going to specify the separator. It is going to be a space, okay, and then the header should be set to truth, which means that it's going to recognize the very first line as the header. So I'm going to hit enter now. So I'm just going to do a head and then GWAS to see if I'm going to hit tab now. GWAS table. It was created. You see here that the first line is called CHR and then SNP and then base pair and then the p-value. Okay, these are the variables or the columns. Okay, so we, and I'm going to ask R, which class does this GWAS table belongs to? It's of the class data frame. Okay, so the GWAS table is a data frame object. You can take a look. Yeah, I already did the head command. Let's now do n equals 10. Why is it complaining? Oh, because I put it in the class. N equals 10. There you go. So the 10 first lines of this object. So let's now plot some figures using this data frame. We are going to use this plot function, this box plot function to visualize the data. In the y-axis, we see the genomic coordinates, and in the it's the opposite, right? In the x-axis, we see the genomic coordinates, and in the y-axis, we see the p-values of the biological association that I just discussed. There's going to be an error there that maybe you guys are going to run into. Let's try. I'm gonna just copy this first line here, see what's going to happen. So I'm gonna ask it to plot. So it plotted the figure. So plotting, and then it's taking the data argument. It's the object that we just created, the GWAS underscore data table. And then the very first argument is what goes in the x-axis, right? It's the base pair, this column here. And then the GWAS underscore table is the p value. It's this column there. So that's why, again, we are seeing the base pair in the x-axis and the p-value in the y-axis. I marked here that it complained, but it did not complain. Is this any different? It doesn't, I do not see any difference between the previous one and this one. So I'm going to go to the next one. Okay, this is just another syntax that I can plot. I can use plot and then the x and the y axis directly. It also accepts that. I can do something different. I can use the box plot function instead of the plot function. And again, we are doing the data equals GWAS underscore table. And X is going to be the base pair, and the Y is going to be the p-values. So I'm going to copy that. I'm going to paste it here. I do not see the names in the x-axis or the y-axis, but I think it's compatible what we see here in the y-axis because y should be the p-values, and here I am seeing 0.0. For example, in the first line, it's 0.01335. This is actually oh, but the thing is that in the y-axis we have minus log of the p-value at the base 10. That's what the difference is. Okay, let's go to the next one. Compare the chromosomal coordinates and the values of the p-values in the box plot above. Compare the chromosomal coordinates. The thing is that in the y-axis, unless I'm reading this not correctly, in the y-axis, The y-axis is base pair. The y-axis is a base pair. No, sorry. The x, well, this is telling me that the x-axis should be the base pair, but this figure, I don't think in the x-axis here, unless I'm reading this incorrectly. In the x-axis, I do not see the chromosomal coordinates. Oh, so is what this is telling us is that in the y-axis, these are uh, the base pair positions. That makes sense because the base pair positions are in millions, and this is 1.5 times 10 to the 8, which means 100 coordinates 150 millions. That makes sense. But then what is, it doesn't make sense because then the x-axis is not making sense to me. So I'm not going to spend a lot of time to understand this. So that was the first point here. This next point here is saying that the chunk below allows us to omit the NA values in the data frame. I'm not entirely sure if there are. From what I remember, there are NA values. Okay, so we ran that. This is creating a variable that's going to highlight the colors of the SNPs that we want to show, and we can just copy this. These SNPs are the SNPs that we want to, I don't know if the paper is not, I do not have the paper open anymore, but the SNPs that we want to highlight in green. Okay, so I copied that. Let me see if it's going to work here because this is huge. Oh, it plotted already. So let me see. Let me go back here. I only need this part here. So it worked, but I want to show you exactly what to do. So it's this command that so remember when I said, so snip highlight is going to be a vector, okay? And the way that you create a vector in R is by doing the C, open parentheses and close parentheses, and what is in between is what the vector is going to be composed of. Okay, so I can now find this vector. So it is created here. So I can always type this command ls, and I can see this object here, right? The snip highlight. It is there. So this object was just created, okay? And now we can finally plot the Manhattan plot. I can't remember creating this GWAS underscore table underscore omit, but here it is. It's that object that I created using the na.omit function. Okay, so let me check if GWAS underscore table and GWAS underscore table underscore omit have the same dimensions. So GWAS table 3624 SNPs. Let's now see the GWAS underscore table underscore omit. Yeah, so apparently there's no need to use the na.omit command because they have the same dimensions. There's no SNPs that were being that had NAs in the columns. Okay, so I'm going to just copy this. And there you go. We have plotted the figure of the Manhattan plot. We have plotted the Manhattan plot. So the Manhattan plot shows the significance of a number of SNPs by showing the coordinations of the coordinates of the SNPs in the x-axis versus the p-value, which means the number of a value that measures the significance, a value that measures the significance level, in this case minus log 10 of the p-value. Okay, so in our case here, these green dots are the SNPs that are located near the sortilin, one of the sortilin genes that I just showed you. All right. Let me see. I guess we're about to be done. Yep, we're actually done. Thank you for watching. See you next time.